Good evening, guys.
Good evening, ma'am.
Do my voice is audible to you, everyone?
Yes, ma'am.
Okay. So let's wait for a couple
Yes,
minutes till the people are joining. Okay. Guys, if you feel any lagging in my voice or PPT do not change, please respond me immediately. If my voice is not perfectly audible also, please respond me so that I can rectify the problem.
Okay, ma'am.
Okay, once again, good evening, everyone. A warm welcome to you for this 7 days workshop. Here we all are gathered to learn and experience the hands-on demonstration of BB, which is called as Beautiful Bioinformatics. So anyone have you heard the webinar which we have provided recently? Anyone? Have you attended a webinar regarding this introduction of bioinformatics?
yes, ma'am. I have attended the webinar.
Okay. Only one? What about remaining? So, have you understood the concept on the courses we are going to provide and remaining everything about that? So, guys, please interact with me, whatever I'm saying, whatever I'm asking the questions. This is a workshop we have gathered to learn more new things. Without interaction, you can't learn anything, right? So, whatever, just feel free to ask me any queries regarding the sessions or any PPT. If you missed the single point, also you can ask me. I will provide you. Feel free to ask me questions. Okay. So here we the BioHelp learning platform are to give a bioinformatics knowledge. So guys, anyone having a basic idea regarding bioinformatics? Do my PPT is visible?
Yes, ma'am.
Okay. So have you heard any time this bioinformatics term?
Yes, ma'am.
So can you say what you know a little bit in the words?
So, ma'am, bioinformatics is combination of biology, information technology,
Yes.
mathematics, and statistics, and computer science.
Yes. In the combination of all these things, this term
Yes,
is evolved.
bioinformatics. Yes.
Yes. Rest of the people, anything
I don't have that much
as in.
information, but I did primer designing and fast sequencing for my internal assignment in my MSc.
Oh, that's great. So have you done PyMol, Autodoc, everything? Have you know?
I know these terms, but I hadn't did that much because I used only Primer
Okay,
3
that's
and NCBI PubMed only.
oh, that's great. No worries. Here you will learn everything. It completely hands-on experiment. Guys, are you listening from the laptop or else you are using your smartphones?
Ma'am, laptop.
Okay. So in the smartphones also, no worry. But if you are using laptops, you can also browse like me itself. Whatever I'm showing in the screen, you can also directly do in your personal PCs also. So let's move to the topic. Here, what is bioinformatics? So here the mice and humans. Let us take an example. Mice and humans. They both have same 80% of DNA, right? So genes are same. Here, what is the genes? Anyone? What is gene?
Coding sequence of any DNA from where we
Yeah.
can get protein, which is functional.
Yes. Exactly right. So how they are organized and how to make the sequence and how to diagnose and a particular disease on a particular diseases how we can diagnose, and all these large data sets need a powerful tools, right? So that is called as bioinformatics, to put all the large data in the one mixture of point in the one ball. Then this bioinformatics have been evolved to know all such things, how the DNA is evolved and what are the sequences, what is the sequence present in there. The powerful computers store and manipulate the DNA, right? So, and the people behind the computers and bioinformatician scientists who are often trained both in biology as well as maths and the computer science, as you all know. So these multidisciplinary researchers develop methods and software tools to program computers to dig through and make a sense of all this data. How the mice human genes are similar and protein coding sequence on this way. In the combination of all these things, this bioinformatician approaches this type of term, like small matching sequence like ACTTGC. These are the sequence, right? Here doing a while doing this bioinformatics, you people will feel like playing a word game in your mobiles like that. So we get a similar sequence when we search any sequence in the PubMed or else in the NCBI, whatever. So when you search any sequence, you get 30% similar sequences, right? So first sequence, suppose we take an example of Margo virus in the VP35 protein. So if you search VP35, there you will get number of articles and number of protein sequences you'll get. So you will select a particular sequence and you'll start performing the activity or else researching on the particular sequence, then you'll get more data. So to analyze everything, to analyze such DNA sequences, this bioinformatics term will become. So by this we can find how the genome sequence have been evolved and how the genes are present and what are the sequences. So to put all the information in the one particular way, this is the bioinformatics we call. So development of technology here, from the mobiles to the smartphones and from the tabs to the laptops, how this technology is developing. Same like that the bioinformatics also. Before if you want to search anything, you want to do research on anything, then we need, it takes some years of time. But then the bioinformatics complete fast travel. So you want to learn ABC, directly you are going to those things.
Excuse
So
me, ma'am, your voice is breaking.
Yeah,
my
ma'am.
voice
Same
is breaking.
Yes, ma'am.
Okay. So what about now? Is it okay?
Now it's okay.
Still your
Yeah,
uh,
it's okay now for me.
yeah, okay. So if you feel like that, immediately respond me, because I don't know whether the technical issues is coming or not. So that if you feel any questions, you can immediately respond me. Am I going?
Ma'am, again, voice is breaking off.
Okay, ma'am. So, what about now? Is it okay?
Yes, ma'am.
Okay. So, development of knowledge. Here, from the mobile phones, to the tabs, and from tabs to the laptops, here we came across like this. So, same like that, the generation is also developing. How the technology is developing in the point of biological way, also the technology is developing. Means how they are developed. Means like example smartwatch, you all guys are very much familiar with smartwatch, right? When you put a smartwatch to your hand, then you will get a sense of your heartbeat and how much activities you have done and how much steps you have put forward and what is the temperature of your body and what is the oxygen levels, everything. Means how this technology is developing. Before, if you get any sick, we'll go to the hospital, we'll go to the clinic and we'll call to the doctor and we perform all these things. But here by smartwatches only, you are going to learn what is the temperature, what is the activity, what is the heart rate, everything. So like that, by this the technology is developing. So based on that only, this bioinformatics is also another platform for the development. So here, the bioinformatics is the application of information technology to store and organize and analyze a vast amount of biological data. So by seeing this first statement only, you can understand it is an application of information technology. The stored data is available in the form of sequences and the structures of proteins and nucleic acids and all in the combination of information. The biological information of nucleic acids is available as sequences, while the data of protein is available as sequences and the structure. So if you want any protein, suppose insulin is a protein, right? So you need a protein structure. You'll go to the Google, you'll search insulin protein and you'll get some images. So how these images are done, how the different colors are available, how they are making, and what is the sequence is present in particular protein. And if you remove one sequence, do that protein get disturbed or not, everything you will learn in the coming up slides. I mean, in the next workshops, in the next day workshops, regarding how to use the pymol. In the pymol software, you will learn what is a sequence and how to give the coloring of a particular protein, and all those things you will learn from this bioinformatics itself. This bioinformatics is a platform for the people who are interested to go to the software side. Means most of the people think after taking BPC we can't do MBBS. If we don't get an MBBS seat, so what we have to do to skip the mathematics subject, many of the people will take BPC. So I think you people also do such thing. So to avoid mathematics in the subject, we will take an BPC and biological and the special interest also, it is mandatory. So to avoid, but most of the people will think they want to do applications and they want to do a software side, they want to go to a software side. So to all such activities, they will select this particular bioinformatics topic. And the sequences are represented in a single dimension, whereas the structure contains a three-dimensional data of sequences. Here, moving to the next slide. Here, by seeing this pictorial representation, have you understanding any of the concept?
Slide is not changing.
Okay, 1 minute. So till now, the development of technology slide is only present.
Yes, ma'am.
Okay. What about now? So till now, I have said about the slide itself.
Yes,
So do
ma'am.
the slide is
Exactly.
visible?
Yes, ma'am.
Okay. So whatever I have said till now, it is present all these things. Bioinformatics is an application of the information technology to organize and analyze a vast amount of biological data and to store the data which is available in the form of sequences and structures of proteins, nucleic acids, and all these things. By seeing this slide, you people can understand what is just it is an introduction part, and this session is completely based on introduction itself. The people who are don't know a little bit about this bioinformatics word, this is a complete introduction. After from the next day itself, you will have the demonstration classes. So yeah, this is only the introduction part. If you skip also, we will provide an recorded sections also. If there's any point, you can go through that. Here, the next slide have changed.
Yes, ma'am.
Okay. So by seeing this pictorial representation, are you getting any idea regarding this? Anyone?
Yes, ma'am.
Yes.
Ma'am, the
Can you?
gene sequence is converting into uh, uh,
Yeah, you are right.
machine understandable binary language, that is.
Yes. So whatever the sequence which is in numeric, which is in alphabetical way, when we started doing research on the particular sequence, or if you are doing any drug discovery on a protein modeling, then the sequence will be converted. So how the sequence is converted, all these things you will learn in the softwares. You can also change the sequence, and if you want to, you can also replace the molecules according to the performance or according to the functioning of a particular protein. So the numerical data, we can't keep all the data in our brain, right? To feed on a particular sequence, we will put in the system in the personal pieces. Then you will understand what is a sequence, and which sequence is from which protein, and what is a protein name, and what is the structure name of a protein, and all these things you will learn from the bioinformatics pathway. Here in the next slide, so by seeing this slide itself, you can understand in the combination of biology and the computer science and information engineering and mathematics and statistics, in the combination of all these things, this bioinformatics term has been evolved. And here this is a pictorial representation. You can see the laboratory, how the bioinformatic laboratory is present and all. Here in the next slide, here the DNA simulations is there. When you took a sample of DNA, then you will get a DNA structure, and this DNA you will identify the sequence. And when the sequence is put in a particular when you feed the information in the PC, then you will get in the numerical form, then the algorithm simulation, then you will get a result. So like that, this is the process how the DNA simulation is taking place. I think in your undergraduates, in the masters, you will learn all these things, how the DNA simulation is taking place, and how to demonstrate the DNA, and how to identify the DNA molecule, to how to identify the nucleotides from the DNA sequence, and all these things. Here this is a just pictorial representation, how the DNA sample is evolved and what is the sequence. Then that sequence will put in the PC, then you will get an algorithm simulation, then you'll find out the result. So like that. So areas where bioinformatics is applied in the genomics, as you all know, genomics means the DNA, RNA. Genomic features prediction and sequencing data analysis, and all these things comes under the genomics. And also proteomics, according to the point of view in the proteins, the you can also see the 3D structure. And also if you want to do any drug discovery projects, if you are doing any project regarding a drug, on a particular drug, if you want to create a new drug, recently we have faced COVID-19, right? So to create a vaccine, we need some proteins, and also we need some DNA sequences and all, to learn how the protein 3D structure is modeled, and how to do the protein structure modeling. So from where these proteins have been evolved, from which species, specimen or species we have to take that particular protein, and all these things will come from the proteinomics. And the system biology, the gene pathway enrichment, pathway analysis, and in the point of view, in the phenotype, image analysis and integration. Here in the pictorial representation you can identify the system biological means, in such a way, the glycolysis and how the lipid molecules is the taking place, protein molecules, and all these things comes under the system biological. Pathway analysis, how the particular, when we put inside a particular medicine, how they are traveling, how they are moving. So to learn all these things, in the system biological also, this bioinformatics is applied in the proteins, in the genes, in the phenotypes, and all these things are in all these areas. This bioinformatics will be get applied. So moving to the next slide, anybody can do it. So most bioinformatic analysis can be done by using web applications and commercial program with GUIs. So here this is a NCBI. I think anyone have an idea regarding NCBI and PDB?
In
No, ma'am.
NCBI, we can retrieve the sequences of nucleic acid, proteins, and other things. In PDB,
Yes.
we can retrieve only the sequences and structures of protein.
Yes, we will get exactly what is a protein structure, and what is a how the protein structure, how the protein molecule is made, and what are the nucleic acids. Not only protein, you can also identify the brief structure also. When you download the faster sequence of a protein from the PDB, then you will put it on the Autodoc Vina, or else in the PyMol, or else in the MGL tools, Rasmol. These all are the softwares. When you put on this, you can observe the exact structure of protein, how the protein molecule is in the 3D format, in the 2D format. From this protein data bank, you will add. Here there are number of articles are present in the protein data bank, and in the here in the NCBI page also
Ma'am,
you can identify. Yeah.
ma'am, from this library we have to download the protein and then put into the software, right?
Yes.
Okay.
That is your question.
Yes, ma'am.
Okay. So, in the future, you people who are interested doing research in a particular drug, like example, as I'm doing on the Margo virus on the VP35 protein. So we have to go to the protein data bank, and we have to search in the search bar of a particular protein, like whatever we need, a protein of interest. We have to select. We have to put a name on search bar in the protein data bank. Then when you click on the search, then you'll get a protein structure. From that protein structure only, means whatever, if you want to do any project, first you people should need a literature review. Means when you read number of articles only, you can identify, because a particular disease not only evolved in particular day or no. So in the previous, from the since 90s, from since 80s, the particular disease have been evolved. From there to till now, how this disease have been evolving, and in which state, in which season that the diseases get down, and in which the disease is raised, and which type of human beings this disease will be get attacked. Means the diseased people get more attacked than compared to the healthy people. So all these things comes under, you will learn from this only, from NCBI. So when you search any protein from the protein data bank, that protein molecule you will keep in the software. The software also you will learn from the next slides itself, in the PyMol. That protein molecule exactly, you will put it on a PyMol software or the Rasmol or whatever the softwares you are using. It's based on your complete interest. So when you put, you will get some sequence. So in that, from that sequence only, you have to start working on it. If you see unnecessary protein sequences, some sequences will be like, this sequence, there is no use of it, some particular template, so you can also remove. So all these things you can identify from the PyMol software. Means you have to create your own protein. So from the PDB, when you download the protein molecule, that protein molecule, when you put it on a software, from that protein molecule only, from that protein only, you can create your own protein. Means some proteins will be like in the cluster form, like a completely big structure. So if you are creating something new, if you want to remove unnecessary sequences, also all these things you will learn in the software side. So that is the main thing is to know to see the structure of protein. This protein data bank homepage is necessary. This is a software. You can also download the protein sequences, protein structures, and all these things. So NCBI is nothing but the abbreviation is National Center for Biotechnological Information. So you can read many of the articles, you can submit or you can download the articles, and you can see the BLAST sequence. Can anyone say the abbreviation of BLAST? Have you heard term BLAST? Anyone? Am I audible?
Yes, ma'am.
Okay. So anyone? BLAST? Yeah, basic alignment local search tools. Yes. Okay. So BLAST also, and also you can see the protein, and from the PubChem you can download the ligand molecules, gallic acid, all these things you will learn in the next slides. No worries. So moving to the next slide. Bioinformatics is quick, right? So bioinformatics analysis can be done quickly because computers are involved. So no need to go to the lab wearing the apron, using the gloves, and start doing simulation, taking the sample and put it in the incubator and observing for 24 days. Some medicine, some drugs, some proteins will be visual till it takes some years of time, some months of time, all these things when the humans are involved. But compared to the humans, when the computers are involved, this bioinformatics is a term which became very fast. You can also do very quickly. When you enter the data, when you click the enter button, then you will get the complete information on a particular search. Whatever you searched on a search tool, then you will get the sequence, everything. So this bioinformatics is very quick. So the three things should be remembered. Had the slide changed? Okay. So three things to remember. Bioinformatics requires dedication and continuity. So guys, keep remember, if you want to do any project, research article, if you want to write any article on a particular drug or else on a particular disease, whatever, but it should be complete dedication and continuity. So after doing all these things, at a final point, when you miss something, any sequence, if you miss, and the complete result will be down, so you don't get an whatever the expected result. So you should not stop your activity till there. So I did not get the correct result, so waste of time, I have wasted much time, so I don't want to do these things. So you should not think like that. You have to keep complete dedication and continuity. Whatever, if you get a result wrong, then start doing again and again, that finally you'll get, the whatever the final point, final research term, right? So you can do continuity. You should not stop in between the experiments when you fail. So bioinformatics data analysis is a full research experiment in itself. As you all know, we get the most out of our research if we work as an interdisciplinary research team throughout. If we work in combinedly also, you will get a exact research. Whatever the research you are doing, you'll get a work and you will get an exact result whatever you are searching. So first, hypothesis. Here in the term hypothesis means whatever you are doing, interested in the whatever. If you are interested on a particular drug discovery or a particular project, first you have to need the term. What is a term? Means where, what is this, and from where this, when it is started, everything. Suppose, like, takes an example of coronavirus, COVID. So when this COVID started, and which people they are getting affected, and which type of people. So you have heard most of the diabetic patient are getting effort than compared to the healthy person. So like that you have to do research, hypothesis, final reason, what you have to do the literature review. You have to read many new articles, hundreds of articles. If you read only, you will get a final idea regarding on a particular project. So number of articles if you read, you will let you know the, you will understand the concept. Okay, so this is the about this is happened. So to avoid this, whatever the happened thing, you have to avoid. So to avoid, what are the knowledge you have to put to know all such things, this hypothesis or the literature reviews must and needed. So if you go to the any researcher, first when you ask the researcher what the work you have started, so I want to do a particular work on particular thing, then they will ask you, first they will give 1 month time for your literature review. First you have to know what is the exact word, what is the term, what is the thing you are doing. So whatever you are searching on a particular disease, you have to know more about thing, the past, present, everything. If you have an idea regarding that particular word or topic, then you can go to the further experiment, that is called as literature review. The term, it is very much important to you guys also in your further education or in your further research. The literature review part is most important thing. Before going to do any experiment, you have to know the basic idea regarding on which thing you are going to learn, and what is the exact thing, what is what, everything you have to know before doing that experiment, so that you will get more idea regarding that, and also you can perform with a well knowledge. So these are the three things is compulsory. You have to need a dedication and continuity, and also you have to do the data analysis on full research itself, and mostly teamwork. If you do, you will get an exact research also. So experimental design, and what is the experiment, and you have to analyze, and you want to evaluate, and then hypothesis. It's completely like you can also do all these things. By doing these things, you will understand the concept of whatever you are going to do on the research, the discovering of new things, and all this. So till now you have learned what is bioinformatics, means from where, what are the tools are involved, all this. Here the introduction, as you all know, no need to take much time on this. So I'll just read it. Bioinformatics is a rapidly developing branch of biology and is highly interdisciplinary, as you all know. Using techniques is nothing but using by uh, PubMed and PubChem and PDB, NCBI, all these are the platforms you will learn in the further next sessions. So all by using different techniques. So informatics, statistics, mathematics, chemistry, biochemistry, and physics, all these things. It has many practical applications in different areas of biology and the medicine also. So in the combination of biology, as one of the introduction, one of said that in the combination of all these things, this bioinformatics term has been done. So this is the thing. And here the bioinformatics derived from the computer analysis, as you all know. Bioinformatics is a combination of biological and information. So these all these things are the introduction part. So before going to do all the research, before going to do all the demonstrations, you have to know little basic thing regarding bioinformatics, so that I have put all these things. So from the, you people don't think why this ma'am directly going through the slides and not providing us by our hands. This is completely introduction part. From the last 4 days you will learn how to demonstrate, how to download the softwares, and all these things you will learn in the next sessions. Yeah, these all these things are the just basic things. Bioinformatics is an science concerned with the development and application of computers, hardware and the software, the storage, analysis, visualization of biological information, and all these basic things. So, guys, if you want to note or if you want to take a screenshot, you can take a screenshot because these are the basic definition which is easy to remember, And also, if you have exam on the particular topic, you can also put this information in your examination also to get a good score. So if you want, I will wait for a minute, then you can take a screenshot also. Here, next slide is the softwares. Softwares means till now I have said NCBI, National Center for Biotechnological Information, and EBI and DDBJ. Can anyone say what is the abbreviation of EBI?
European Bioinformatics Institute.
Then what is DDBJ?
DNA Data Bank of Japan.
Yes, exactly. So here this is the homepage of NCBI. So anyone, have you tried by yourself browsing this NCBI?
Yes, ma'am.
This is completely introduction part. Feel free to ask me or you can
Yes, ma'am.
interact with me. Okay. So what about what thing you have searched in this NCBI?
I have designed primer for BCL2 protein and SRY gene. That's it.
Okay, that's great. Then what about others?
Ma'am, I've searched for sequence of certain genomes for genome annotation purpose.
Yeah, okay. Anyone have read articles from this NCBI?
Yes, ma'am.
Okay. Is do that they are helpful to you? Have you gained any knowledge from that?
Yes, ma'am. Gave a
Yes,
little
ma'am.
insight about the current research going on and the prospects of the future researches.
Yes. So till now, I have said that literature review part is must important for every researcher if they are interested to do further research on a particular drug or else particular protein, whatever. So first, literature part is necessary to do. You people get it out, ma'am. You are continuously saying literature, literature, literature. But from where do we get all this literature? Here you can see the term literature, and also you can also download the data, download the particular sequence when you search any of the protein name, or else if you when you search on any ligand, whatever, when you search here in the toolbar, then you'll get complete information in this home page. Then when you click on the particular thing, then you'll get complete information regarding that. So you have to start reading. First thing is you have to know how to read the literature part, means what are the difference. You will see first page, then when you search, when you click on the second thing, then you will see you have to understand what is the difference between the first and second, and what is the changes have done, and how they are evolved, and how the current, in this current situation, how they are changing, and what are the things what you have to change till now, what they have changed, and next in the future what you are going to change. All these things will be comes under this. So this is just home page. We will all let you know in the further next sessions. Here next, this is the EMBL, European Bioinformatics Institute. Here also you can find the gene or protein, not only from the protein data bank, also you can also download the protein sequence, and you can also search the chemicals. And if you want to do any data, if you want any data resources, you can also find here. And if you want to submit your data, you can also, and here you can explore our research, and all these things you will learn from this EMBL. So in the next homepage is DDBJ. This is a bioinformation and DDBJ center provides sharing and analysis services for data from the life sciences, research and advanced science. Here you can also search databases, and you can also submit your data. Same, all these things are same, but using in the different. It's all your based on your interest. So you can also see the supercomputer. You can also download the sequence. You can whatever the articles, the research papers, you can also download from all these things. So today's work, I want to allot you guys. So just whatever the terms just now I have discussed with you, the NCBI and EBI and DDBJ. These three things. After the end of the session, just go through the your web page and search all these things and try to learn something new from this, so that when tomorrow if you are browsing this, you will get an idea. Oh, okay, after this you will get this, so you'll get an basic idea. So try to make a points on the particular home page, at least minimum 3 to 5 lines. Okay. So tomorrow in the starting session, I'll ask you the questions regarding this. Just discuss with me 2 to 3 points is enough. Okay, are you ready with that?
Yes, ma'am.
Yes,
Okay.
ma'am.
So you will learn something new. Uh, what is the ddbj, what are the sequence, you can put your own sequence only here. See, you can put your own gene, you can put your own protein or chemical, you can search whatever, and then you'll get this. So whatever I discussed here, you will identify it or not, you just search it. Then whenever I'm explaining on a particular day about this thing, you will understand all the things. So moving to the next, categories of databases for the life sciences. So in the life sciences, which type of databases are come under here? The sequences. Here the genomic and the mutations, polymorphism, and protein domain, and proteomic 2D gel, mass spectrometry, and 3D structure, and metabolic networks, regulatory networks, babylography, expression, specialized, all these things. So all, you can also mutations means in the genetics you can identify. You can also see the genomic means, as you have learned in the before slide, genomics means in the RNA DNA performance, and what is the sequences, and how, what is the 3D structure of a particular protein, and what is regulatory network, expressions, and all these things comes under the categories of the databases. So literature database. Coming to the next slide. Literature databases means, here you can see bookshelf, a collection of searchable biomedical books linked to the PubMed. So if you want to read any particular topic, then till from the starting of the session to here, I started explaining all these things. What is literature, how the sequence is evolved, and all these things you will learn. And PubMed allows the searching by author names, general titles, and new preview index optional in the PubMed. And PubMed database provides access to over 12 million Medline citations back to the mid 1960s. It includes history and clipboard options, which may enhance your search session. So these all the terms. Please make a note of these terms and search in the Google. Then you will identify the people who are interested to do the further research. These terms will be helpful to you, because to learn more informative, you will get from all these things itself. So online Mendelian inheritance in man in the database of human genes and the genetic disorders. If you want to do a research in the genetic disorders, and this is a platform, and here this is a platform, you can see all these things. 1 minute. Okay. So online Mendelian, have you noted these points? PubMed, OM,
Yes, ma'am.
okay. So till now we have completed all the introduction part, exact the term, what is bioinformatics, from where this bioinformatics is applicable, and what are the things involved, and what are the softwares, all these things, not the softwares, but the home pages of NCBI on all those things. Here you will learn next rest of the things. Here the term bioinformatics tools. Here the DNA sequences. So for the proteins, for the DNAs, for the RNAs, for the three, you will have different type of home pages. And here this is for DNA home pages, and this is proteins and some bioinformatics. Okay, I'll explain slowly. So DNA sequences, they are the big databases. Do this slide is visible? DNA sequences.
Yes, ma'am.
Okay. So they are the big databases, and searching either one should produce similar results because they exchange information routinely. So Gene Bank, NCBI, as you all know, this is a logo of NCBI, and here this is the DDBJ, DNA Data Bank of Japan. And if you want to do research in the East, if you want to collect some information about the East, and here this is the website, and some of the specialized databases for tissue spaces, that is called as expressed sequence tags, and NCBI, Trigger, TIGR, and many more. Not only this, you can also see all these things. So I'll give a couple minutes. Please make a note of all these things so that it will be easy to you. If you want to do a DNA sequence, then you will directly go to the search of GenBank or DDBG and all. I'll give a minute. After completion of that note, please respond me. I will change the slide. Or else you can take a screenshot also. Have you done?
Yeah, ma'am, done.
Okay. What about others? No one is responding. Please interact me. The interaction
Yes,
of
ma'am,
you
done.
only you'll get information. Okay. Only two people are responding me. What about others?
Done, ma'am.
Okay. So moving to the next slide. Protein databases. Here this is also a big databases. To search a protein molecule, here these are the softwares you will use. One is a sweet spot, and here this is a website, and this is a logo, and PIR, protein identification resources. Please make a note, then you can identify easily. The world's most comprehensive catalog the information of a protein you can identify from the protein identification research resource. And the translated databases, translated EMBL, including entire that have not been annotated yet it into the switch code. Compared to the switch code, EMBL also mostly used homepage, and GenPept that and the PDB, as you guys are very much familiar with this. The sequences you can derive from the 3D structure. So for this also, I'll give a couple of minutes. You can just make a note of all these things. If you are done, you can respond me. Have you done?
Please wait, ma'am.
Okay. From tomorrow sessions, please be fast with your book and pen. So whatever the words it is different to you, please make a note, and you can also do the search in the Google, and you can also ask me in the next sessions also. So please make a note each and every point which is difficult to you. So can I move?
Done, ma'am.
So can I move to the next slide?
Yeah, ma'am.
Okay. So some bioinformatics tools. Here, this is also some tools. Blast, which is very much familiar, basic local alignment search tool and alignment tool, and GRAIL, gene recognition and analysis of internet link, and primer designing tools and all. Please make a note on this also. Have you done?
Ma'am,
Please
slide
make
not,
fast.
ma'am, slide is not seen yet. It's not visible, ma'am.
Okay. Do my slide is not visible to you?
No, ma'am.
Okay. Now, is it okay?
Yes, ma'am.
Okay. So make it fast. Just take a screenshot of this so that it will reduce our time.
Done, ma'am.
Okay. So bioinformatics applications. Molecular medicine. Coming to the molecular medicine, the human genome will have profound efforts on the field of biomedical research and clinical medicine. Every disease has a genetic component, right? This is a new knowledge of a molecular mechanisms of disease which enable a better treatments and cures and even preventative tests to be developed. So the term bioinformatics means these are the applications in the molecular medicine point of view. Also, this bioinformatics is applicable. In the point where it is means clinical medicine also, like coming to the COVID-19, here we have developed something new medicine, right? So fast sequencing, we have done the sequence, we have selected the proteins, which proteins are involved on a particular disease, and we started doing that, we have started doing performing on the active research on that. Then you will identify the clinical medicine, you can create some clinical medicines in the bioinformatics also. The human genome will have profound efforts on the field of biomedical research and the clinical medicine. Every disease has a genetic component. This is a new knowledge of a molecular mechanisms of a disease will enable better treatments and cures and even preventative tests to be developed. In the molecular medicine point of view also, this bioinformatics is applicable. And coming to the drug development, at present all drugs on the market target only about 500 proteins. With an improved understanding of a disease mechanisms and using computational tools to identify and validate new drug targets, more specific medicines that act on the cause. not merely the symptoms of a disease can be developed. These highly specific drugs promises to have fewer side effect than many of the today's medicine. So whatever, if you are doing any research on a particular disease, then you have to keep remember all these things before going to develop any drug, or if you are doing any drug discovery new thing, then it should not give any symptoms other than the disease cure. And it should not whatever the drug you are using on a specific, that should not provide you any side effects. And also whatever the target computational tool which you are valid a new targets, then the specific medicine act of cause which is should not give any other symptoms. So it should not give any symptoms whatever the target. Suppose example, when we are getting fever, then we will take a medicine of paracetamol or dolo 650. Then when we put inside, then the fever will be cured. But do we get any other diseases from the dolo 650 by using that drug? No. So we don't get. So it should not, whatever the drug you are discovering, it should not cause other symptoms. It should